Hello everyone, today we continue to study muscle tissues, and as usual we have a lot of work in our practical book. We have a lot of laboratory works or just watch videos to them, and as usual I will leave the links to this practical, and it is a lot of channels, a lot of video files. And let's start. We have already studied all the structures of the muscle fiber and even discussed the muscle contraction. I just recall the stages of muscle contraction. This will help us understand following issues. Contraction of muscle is connected with the appearance of the action potential on the membrane of muscle fiber. Thanks to the synapse, action potential can conduct on the sarcoplasmatic reticulum and move inside of muscle fibers. You see this place, muscle synapse, and the mediator acetylcholine, and thanks that the calcium ions can contact with the special protein troponin. You see the calcium ions. And then after that results in this interaction of the actin and myosin fiber is removed. Heads of myosin are attached to actin and perform the pull, and results sliding of actin between myosin appears. In the muscle fiber, ATP molecule is used for the following process. You see the ATP molecule, it is just an energy inside of muscles, and use the work of sodium-potassium pumps of the sarcolemma. Thanks to this mechanism, the excitability of the muscle fibers is ensured. Next one, movement of cross bridges of myosin. Myosin heads contain the special enzyme for ATP molecules, and thanks to this the process of contraction is actually ensured, and actin filaments slide along the myosin filaments. And operation the sarcoplasmatic reticulum pump. Thanks to this mechanism, the process of muscle fiber relaxation is ensured. All of these activities require energy. Many interesting facts about energy. About 40% of ATP is used for contraction, the rest for just production of heat. Muscle use the stored ATP molecules, but it supplies enough for 4 or 6 seconds of work, and then you need to turn on other mechanisms for obtaining energy. And next slide we see this process, how many different molecules can use the muscle for production of energy. After using the ATP reserve, these molecules must be synthesized. First of all, we need to know which molecule we will use and to which way, aerobic or anaerobic, we will go. Source of energy in the muscle, creatine, first molecule, creatine phosphate, glycogenolysis, anaerobic glycolysis, oxidative phosphorylation, fatty acid oxidation. So much ways we use to sum energy. As you can see, each path has its own duration and goals, and many molecules can be used to generate energy. Let's start with the creatine molecules. It's very important. Creatine phosphate. The creatine phosphate molecule, as you see, and its pathway is the most powerful, but it needs oxygen, not the right way, to restore the level of phosphocreatine and normal blood flow. We need a lot of blood flow with the oxygen in hemoglobin protein, with contact with hemoglobin protein. So we need blood flow, oxygen, and for production of energy. It is enough for a few seconds. Then phosphocreatine turns out. The next step is the anaerobic breakdown of glucose. You see here it's not so much creatine and creatine phosphate molecules inside the muscle fiber. So next one, we need to glucose pathway, glycolytic pathway, or lactate or anaerobic pathway. It's next 15 seconds of work of muscle after the start of contraction. It does not last long, and at the same time lactate is formed. Pyruvate turns into the lactate and into coenzyme A, which does into the Krebs cycle. Accumulation, yes, here you will see this a lot of these molecules in the glycolytic pathways, and how what way we can use these molecules. Accumulation of lactate in the muscle can lead to the changes in a pH and negative consequence, of course, and we should always move out lactate in the liver, and here you see we just move lactic acid some ways in glucose. Therefore, it's necessary to constantly dispose the lactate in the liver, and it needs blood flow too, and especially we need to move lactic acid inside the blood vessels and from the blood vessels to liver. It's necessary constantly. The most profitable way, of course, aerobic way. So we need a lot of oxygen. It's next way to have energy inside the muscle. It is aerobic mechanism. Glycolytic turns on after 1-2 minutes after start to work and gives the largest number of ATP molecules inside the muscle fiber. And now that we know about the energy of muscle contraction, let's look at the types of muscle fibers. It's very interesting what kind of muscle fibers have what kind of way what way we have the energy in these fibers. Let's look at the types of muscle fibers. They are sometimes divided into just red and white fibers. You see on the photo, but I will be more correct to classify them according to the type of energy process. Red, slow oxidative, medium, fast oxidative, and fast one, it's fast glycolytic, white color fibers. And you see special sign for these fiber types, and you see different capacity for these fibers on the table. All of them have their own characteristics, and so we meet another important molecule. This you see, we can see myoglobin molecules and description for all these types. About this very important molecule, I will leave the links, and you know, inside the red blood cells we have special protein hemoglobin. This Protein keep oxygen and move through the blood flow, but inside the muscle we have almost the same protein, but it's better to watch the video about to these molecules for understanding how much energy we can have in different types of muscle fibers, which contain more or less these myoglobin molecules. I will leave links to the video, and different types of muscle fibers are involved in contraction differently. It depends on the intensity of the exercise. You see the slide. Depend on the intensity of oxygen process. We have different activity. Next slide. Depending on the type of exercise, two modules of contraction of the isolated muscle are possible. Isotonic contraction. This type of contraction occurs when a muscle moves a load, and one of the muscle ends is fixed. One more slide, please. Fixed, and other is free. Why free? Because the weight is not so heavy, connected to the load. Isometric contraction. This type of contraction occurs when both ends of the muscle are fixed, so heavy load. Muscle force is determined by the maximum load that the muscle is possible to fit even a little. We can use metric, centimetric, or we can use a millimeter. You see, muscle force is determined by the maximum load that the muscle is possible to lift even a little. Factors that affect the stress of muscle contraction. Here we see at the molecular level the force of muscle contraction depends on the number of complexes between actin and myosin molecules. In turn, there is a dependence on the primary length of the sarcomere and number of calcium ions and even ATP molecules. At the cellular level, force depends on, next slide, depends on of the number of myofibrils in the muscle fiber and how they are located. The more myofibrils are included in the contraction, the greater the force. Distinguish between maximum and absolute muscle stretch. The maximum force in isotonic model is determined by the maximum mass of the load that the muscles can lift in kilos, for example. In isometric model, it is determined by muscle tension. Therefore, when talking about muscle stress, always we are talking about the maximum voluntary muscle stress, the total volume of isometric tension, more precisely the total moment of a muscle group with the maximum voluntary effort of the subject. Absolute muscle stress is the ratio of the maximum force of isometric contraction to the area of the physiological diameter, kilo per square centimeter. The physiological cross-section considers with the geometric one only in a parallel fiber muscles. Muscle with the oblique fiber arrangement have a large physiological cross-section and therefore greater force. There you see maximum and absolute muscle force, and there the maximum force and absolute muscle force. So this kind of muscle are more force than this one. Next slide 3. At the organ level, force is due to the spatial and frequency summation of nerve impulse. Spatial summation is an increase in contraction force due to an increase in contraction units. Here you see frequency summation. Tetanization is an increase in the force of contraction due to an increase in the frequency of nerve impulse. Here I will link this video to this video. Here you can see the frequency of situation and force and all mechanism of this. So let's look at the frequency of stimulation of motor neurons, and a single action potential from the motor neuron will produce a single contraction in the muscle fibers of its motor unit. This isolated contraction is called a twitch. A twitch can last for a few milliseconds or 100 milliseconds depending on the muscle type. The tension produced by a single twitch can be measured by a myogram, an instrument that measures the amount of tension produced over time. Each twitch undergoes has 3 periods or phases. The first phase is the latent period, you see, during which the action potential is being propagated along the sarcolemma and calcium ions are realized from the sarcoplasmatic reticulum. This is the phase during which excitation and contraction are being coupled, but contraction has yet to occur. The contraction phase, this pink side of the picture, occur next. The calcium ions in the sarcoplasm have bound to troponin. Tropomyosin has shifted away from actin binding site. Cross bridges formed and sarcomeres are activity shortening to the point of peak tension. You see this place. The last phase is the relaxation phase, when tension decreases as contraction stops. Calcium ions are pumped out of the sarcoplasmatic reticulum through the sarcoplasm, and cross bridges cycling stops, returning the muscle fibers to their resting state. Also, a person can experience a muscle twitch. A single twitch does not produce any significant muscle activity in a living body. A series of action potential to the muscle fibers is necessary to produce a muscle contraction that can produce work. Normal muscle, you see here, normal muscle contraction is more sustained, and it can be modified by input from the nervous system to produce varying amounts of force. This is called a graded muscle response. The frequency of action potentials, nerve impulses from a motor neuron, and the number of motor neurons transmitting action potentials both affect the tension produced in skeletal muscles. And the fibers are stimulated. While a previous twitch is still occurring, the second twitch will be stronger. This response is called wave summation, because the excitation, contraction, completing effects of successive motor neuron signaling is summed or added together. At the molecular level, summation occurs because the second stimulus triggers the release of more calcium ions, which become available to activate additional sarcomeres while the muscle is still contracting from the first stimulus. Summation results in greater contraction of the motor unit. You see more and more and more. If the frequency of motor neuron signaling increases, summation and next muscle tension in the motor unit continues to rise until it reaches a peak point. The tension of this point is about 3 to 4 times greater than the tension of a single twitch, a state referred to as incomplete tetanus. The way summation and this incomplete tension, you see. During incomplete tetanus, the muscle goes through quick cycles of contraction with a short relaxation phase, very short, for each of tension. Of the stimulus frequency is so high that the relaxation phase disappears completely. Contraction becomes continuous in a process called complete tetanus. This one, complete tetanus, this feature. If we talk about contractions, then the muscle is not completely relaxed. It is in tone. Exactly, all muscle always in tone. Next slide, you see self-regulation of muscle tone, and I will leave the links about all control. We have already particularly analyzed this issue in previous practical. I will leave the links to the video, and that will help you understand this topic in more detail. But next one slide, you see this. You need to remember what a muscle spindle is. So we will study the high centers of regulation in the central nerve system section. We will study this about all this process after we can understand what is this spinal cord and what is this nerve centers. Let's move on the second part of our practical work. It is the practical work 20. It is single muscle contraction and tetanus. You can read the text distribution in our practice book on page 74. I also leave a link to the video that describes the tools to this lab works. Thanks YouTube channels, a lot of YouTube channels which can help us in this. Let's watch the video of lab works about twitch and incomplete tetanus and complete tetanus. We use a special tools. It's named chemograph. Chemos, it is a wave. We can have the waves during the muscle contraction. Just fix the muscle in a special equipment and contact with chemograph with a hook. Use the electricity for stimulating of muscle, and you see here, you see twitch, single muscle contraction. This part contraction and relaxation, latent period, contraction, relaxation. We have the myogram. And now we have uncompleted contraction, uncompleted like a series of twitches. Thanks to summation of action potential, if we take the more gigahertz of stimulus, we have complete tetanus, and complete tetanus, and complete tetanus. Here you are. So we use almost 200 gigahertz in this lab works. It's this optimum of action. After that, it is the maximum of force. It's like a negative reaction of the muscle on the very, very high electricity. And one more, we just have some examples. One twitch, it's incomplete tetanus. And now complete tetanus. As you see, this chemogram for different action on the muscle. Now I want to show you the difference in the work of skeletal muscles and smooth muscle, and I just we take the part of digestive system of the frog. It is the smooth muscle. It is part of the wall of digestive tube, and let's watch the video. This muscle can contract automatically without any irritation, without any electricity stimulus. You see the difference in contraction of skeletal muscle will very quickly work, but this muscle, smooth muscle, contract very easily, slowly. In this case, we will use the liquid acetylcholine molecule on the part of digestive tube, and let's watch what will happen. The force of contraction is more. And the contraction are prolonged. It takes more time. If we use noradrenaline or adrenaline, for example, epinephrine molecule, it just stopped contraction at all. So it is all for this practical work, but we have one more. On page 79, this is a complex work where we will see the ability of the muscles to stretch and take their shape again after removing the load. Unfortunately, I don't have the video to this lab work, so we'll just look at the picture. In this laboratory, we can calculate that work the muscle performs. The muscle will easily lift a smaller mass to a greater height, and with an increase in weight to a small height. You have an example in the table here, you see, and the next there is a special formula here. The work of the muscle is determined by the mass of the load raised at a height. So next slides. Performance of any work leads to fatigue. This is a temporary loss of working capacity. This condition passes after rest. Fatigue is manifested by a decrease in a shortened amplitude, in increase in the latent period, causes of fatigue at the cellular level, decrease in the amount of ATP, accumulation of exchange products. At the organ level, fatigue is fatigue of nervous centers or the central nervous system, and fatigue of synapses. Just acetylcholine molecules can't produce enough. We have work Next work, I found this slide. Muscle and synaptic fatigue on the page 80. I will leave links and you read the description to this work. And on the slide you see an ergogram. This here. Here you see the ergogram. What is this ergogram? Namely, the record of the fatigue process. And I will leave the links, and even we will have the video. Let's watch this video. I will turn it.
calibration part, we have drawn a baseline here, and we have.
So you see, in the end of this lab work, the student can to move and contract the fingers so easily, and the contractions start a bit slowly. And one more lab work for today. It's a work about dynamometry of the hands. Let's watch the record. For this we will use a special equipment tool, And dynamometry, it is this lab work to know the force of hands muscles. You just do press the stool, and on the scale you see how many kilos the force of your muscle. In this case, one hand has 20 kilos of force, and the left hand has a little bit less. And these two for dynamometry of muscles of back. It is 28 kilos for first time and 40 for the second. So on this slide you see the description to the work of 23, and here you can find the link to the video on the YouTube channel, and here photos of different kind of dynamometers. In this video we'll use this kind of dynamometer. So at the end, as usual, I want to show you your homework. Here you see the homework in your book Animal Physiology about muscle physiology, and for next our practical I ask you to read pages about nervous system, and as usual we have tests about this term physiology of muscles. And for your work in your practical book, you should answer the question set of activity 5 and 6. It is all for today. Good luck. See you next time.